I had my volume up.
Yeah, I was in her like first class.
All right. How's everybody doing?
Tired.
What's that? Tired? Who's
Yeah.
tired?
Winston.
Hey, Winston. Why are you tired?
Just workouts. I just got out of the shower, so I'm just tired from that.
Okay. What team do you play on?
Football.
Football. Okay. So have you guys gone and gotten your Apple Pencils yet? Those of you without Apple Pencils?
So I kind of run into an issue with that because I'm out of state right now and I won't be coming back till the 20th, so the Sunday before. And the earliest I can make an appointment is at 9. So, and they
on
sent
the
me an
20th.
email.
Yeah.
And they sent me an email asking me to move my appointment to like 2:15, and I emailed them back saying I couldn't, and I never got a response. So,
So when do you get back in town?
I fly back in at like 5 in the afternoon on Sunday.
Okay, 5 in the afternoon on Sunday. That's when your plane arrives?
Yes.
Okay. Let's see. So yeah, my Apple Pencil has a crack in a part of it. All right. All right, Mackenzie. I'll text you my phone number.
Okay.
On your way back from the airport Sunday, drop by my house, and you can borrow my Apple Pencil for the exam.
Well, I think my roommate has one too, so I might just borrow hers. I'm gonna ask her this afternoon because we have a group project due tonight too. So. Okay.
When you get it, one of the things that you have to do is you have to sync it with your iPad, and you do that by plugging it into the port. It's got this little end on it that fits in the charging port. And then once you do that, it'll be synced with your iPad now as well as hers. But if you just try and use it without doing that syncing, it won't work. So, all right. So just let me know if you need mine, because I will not need mine because I will not be taking a test on Monday.
Yeah. Okay.
Yep.
I can do that.
All right. What about the rest of you who didn't have an Apple Pencil already?
So I have one.
What's that?
I have one, but
You have one?
yeah, but I've been in kind of a unique situation because I'm paying for classes myself. I was trying not to use my iPad so that I wouldn't have to pay $500 for the technology charge.
There's a $500 technology charge? So you get out of that just by not using your iPad?
Sure, I have to go talk to like the business office and stuff, but I got it taken off. I took a break last semester and they charged me last semester for it. And so I talked to them and they were able to take it off. But
check your figures. My guess is if you possess an iPad, they've charged you for it.
Yeah. Well, the thing would be is that if they charge me for it, I was gonna like return it and say
Oh, okay.
not paying for this if I don't
All
have
right.
to use it.
Well, that's gonna make doing this way more difficult.
Yeah.
So let me make some notes for myself. I was a first generation college student who worked my way through college, so I feel your financial pain. What about the rest of you?
My dog ate mine, so I don't have one either.
Your dog ate yours?
Yeah,
I
she
could
sure
hardly hear
did.
Blair, so I turned myself up, and now I just busted my ear bones. Anyway, Alyssa, so your dog ate yours?
Yes.
That sounds like what you do with your homework.
Yeah, I wish that I wasn't lying, but she did.
Okay. So is it still possible for you to have you contacted Heath to see the possibility of renting one for free? I'm not sure what that means when he wrote that in the email from the tech people.
No, I haven't asked him about that. I didn't even know that that was an option yet.
Okay, so I sent out an email.
I
think
just
sent
got my
everybody.
iPad back, so I just got my school email back, so I haven't gone through all of them yet.
Okay. So I forwarded so I contacted Heath on Monday and said, hey, I have some CTI students who don't have Apple Pencils. Is it possible for them to get those? And he responded back, yes, they can rent them for free for this semester. I assume that means like you check them out and then you have to give them back at the end of the semester is what he meant
Okay.
by that. But check with him on that.
Okay.
Blair, if push comes to shove, I can also just print a hard copy of the exam for you, and I'll set you up somewhere over here, and you can take a hard copy version of it. That'll be easy enough to do.
Can I come
Alyssa?
and pick up my food journal today too?
Can you what?
Come pick up my food journal.
Oh yeah, they've been sitting outside my office since Monday.
Okay,
There's
I
still
wasn't sure.
3
I
of them
wasn't there on Monday, so.
Okay. Yep.
Perfect.
Yeah. What about the rest of you? You feel confident taking an exam on an iPad? Okay. All right. Did I talk to you about academic honesty and exam taking last time? Or was that my evolution in ecology class?
Yeah, you talked about it.
Yeah.
Okay. All right. Yeah. So just be on your best behavior while you're taking this, because I'm not prowling the room looking at you. Yeah. So Blair, update me on your situation and we'll work something out. Mackenzie, update me on your situation and we'll see what we have to do. Same thing with Alyssa, and so I assume the rest of you are good to go then.
Hey, I have a question.
Yeah, Marcus, what's up?
She asked a question about food journals. You say you had them outside of your office since Monday?
Yeah, if you turned in a hard copy rather than a digital copy, I just look through them and then put them back outside my door.
Okay, got you. Okay, got you.
Yeah, so that's for people keeping a hard copy.
Got you.
If you turned it in electronically, so on the Moodle site, there's a grade book that's beginning to get populated. I'll put your grades in there. And so basically the food journal is, did you do the food journal? Did you do it on a daily basis? If you did that, then you get 5 points for that episode of food journaling. So, other questions?
Yeah, and then when is the next food journal due, or is that the one?
So those are the dates are on the Moodle site. So it gives you the time span, the time range. It goes from one Friday to the Friday 2 weeks from that Friday. So at the very top of the food blogging prompts, those are also the dates for the food journals. So every 2 weeks on Friday, you turn it in. I try and get it back to you the following Monday so that you can then have 2 more weeks of food blogging.
Okay.
Yep. Or food journaling, rather. All right. You guys have requested a exam review. So I started working on a review sheet for you this morning when I got into work. So I'm going to share my screen with you. So I suspect I'm about halfway finished with making it. But here it is, just so you can see it. When I'm done with this, I'll just post it on the Moodle site. What does the omnivore's dilemma mean? Who wrote about it first? That's a guy named Paul Rosen, in reference to rats. Why are koalas not faced with the same dilemma? This is kind of an application question. I obviously haven't spell checked this yet. What does Michael Pollan refer to when he refers to the United States having a national eating disorder? Contrast the French paradox with the American paradox with regard to diet and health. These are all things that we've talked about in class. Be able to identify the major structures of the plant body, major structures of the leaf. What are the products and reactants in the chemical equation for photosynthesis? In addition to these things, what other things are needed for photosynthesis to proceed? These are things like rubisco and sunlight and chlorophyll. Once again, all things that we've mentioned and talked about in class. What enzymes are involved in initial carbon capture in C3 and C4 plants? Where in the leaf does this occur in C3 and C4 plants? Where in the leaf does carbon dioxide get converted into glucose in C3 and C4 plants? Why is there spatial separation in where this happens in C4 plants? What does this mean for water balance of C3 and C4 plants? Why do the last 4 items in this list result in C4 plants having a higher ratio of heavy carbon isotopes compared to isotope ratio in C3 plants? This is what Pollan talked about in his book, which is how we went down that whole path of talking about photosynthesis. And that's as far as I've gotten because I'm just about to start in on the genetics part of things. I'm gonna probably put some things on there about the distribution of corn and soybeans and wheat and things like that in here as well. But you can see that you know, a review sheet ends up just being, you know, what are the things that we've talked about? It is not necessarily these are things that will be questions on the exam. These are things that could be questions on the exam, but these are the kinds of questions and topics that I expect you to be knowledgeable about on the exam. Does that make sense? Okay, I'll complete this probably in the hour or so after we leave here, and I'll probably have it posted before noon today. So log on, look over it, and if you have questions about anything on there, consider also the fact that we won't be finished with material yet. So at the end of the review sheet, there will be some things that we haven't yet covered because we'll be covering those on Friday, hopefully. And hopefully that will help you. I've had a couple of students from my evolutionary ecology class coming and asking me, you know, about ways of studying and things like this. One of the most productive things that I did when I was an undergraduate in terms of preparing for exams was to try and generate as many test questions of the kinds of test questions that I expected my professor to put on an exam, and then go about the process of trying to answer those questions to the best of my ability. And one of the ways I did this really effectively was in most of my classes as an undergraduate, I had study partners. And when I got into a study group, the requirement of the study group was when you showed up to study, we weren't studying during that time. We had already studied, but what you did when you came to the group was each person was required to come with 10 questions that they generated that week. So we met weekly. Every person came with 10 questions that you generated. Some of the questions were the same across all the people, but there was invariably some variation in which questions were being asked and how they were being asked. And then our study time together was basically working together to craft the best answers to those questions that we could. And as a result of that, oftentimes we would get to the test and we had already asked and answered all of the questions that appeared on the test. And so group study, especially as I see undergraduates doing it at Jewel, is oftentimes not very productive because they haven't prepared beforehand and there wasn't any clear agenda on what would be done during the study session. And so the study session kind of devolves into talking about whatever, and that might be the course material, it might be, you know, what you did this past weekend, et cetera, et cetera. And so when I was an undergraduate, our rules for the study sessions were really pretty rigid. And a couple of the students I was talking to were like, wow, that seems really rigid. I'm like, yeah, but being really rigid really kept us on task. So study groups can be helpful, but they can also be super detrimental depending on how they're structured. And so if you want to study together, I advise studying together, but make sure that you structure it in such a way that it's a really productive time rather than a waste of time. And I found the process of generating questions and then working together to answer those questions pretty productive. Actually I saved a lot of I saved practically all of my notes from my undergraduate career. They're in manila folders. And one of the students asked me to dig some of them up, and I was looking through them the other day, and I actually found some of my study session notes. And it really is just a question generated and then a really long, detailed answer about that question. And so anyway, that's a productive way of going about it. If you have questions about the material, if you're unclear about something, you can always go and talk to your friends, but chances are they're just as unclear about it as you are. The best thing is to come to me and say, hey, I'm unclear about, I didn't really follow this whole C3, C4 stable isotope ratio thing. Can you explain that to me again? And do that. The other thing is start studying now so that on Friday, when I say, hey, do you guys have any questions? You guys can say, yeah, I was studying for the test and I'm still a little unclear about X, Y, and Z. And then not only do you get clarification on that, but everybody else gets clarification on it at the same time. Questions? We've been talking about genetics. Have you guys been following the whole genetics thing fairly well? Or do I need to clarify anything about genetics right now before we move on to what we call a dihybrid cross? Okay, then we'll move on. I'm going to share my screen again so you can see the slides that I'm looking at. Can you see the mitosis meiosis slides?
No.
No. You cannot?
Not like
Are you talking about like the big slide or like all of them together?
Yeah,
You see
right now
all-
they're just all of them up there because I haven't started the slideshow yet.
Okay.
Okay, all right. I don't know, for whatever reason, my brain is moving slowly this morning. Now, can you see the big slideshow? The big slide? What's that?
Not yet.
No.
No. How about now?
no.
No.
No. All right. That's irritating. How about now?
Yes.
Yep.
Okay, all right, cool. Everybody see?
Yep.
All right, good. All right, something about what screen I had it on or something like that. Okay, so we started last time talking about a genetic, let me make sure you can see all of the board. You guys can see the board also? All right, so we were talking last time about doing a cross between individuals when you're looking at just a single trait, but you can look at as many traits as an organism has and work out the genetics of it. And so this next example is an example of doing this with an organism with two traits. So we have round yellow seeds, round yellow seeded plants, and we have wrinkled green seeded plants. And these are, once again, pure breeding plants, true breeding plants. And they are true breeding because they have been inbred for generations because of the closed flower of the pea plant. And so they would have a genotype that is diploid with 2 big R's and 2 big Y's for round and yellow, and 2 little R's and 2 little Y alleles for wrinkled and green. So one gene controls the shape of the seed, the other gene controls the color of the seed. Now, because these guys are homozygous on both sides here, they can only produce one type of gamete, a gamete with dominant alleles for round and yellow, and a gamete for recessive alleles for wrinkled and green. And when these gametes come together, you get the genotype of the offspring, the F1 offspring, and that genotype will produce round yellow plants. And so once again, remember that Mendel referred to these things as dominant and recessive because in the F1 generation, in the first filial generation, the recessive trait recedes into the background and you only see the dominant expression of the trait. This is the parental generation. This is the F1 generation. These are the gametes that are produced by the parents. So whenever you're working a genetics problem, the first thing that you should ask yourself is, what are the genotypes of the parents? Given the genotypes of the parents, what kinds of gametes can those parents make? Once you have the gametes that they can make written down, it's really easy to then just combine those gametes with one another through fertilization to get what the offspring will be like. So we go from 2 phenotypes. Remember that this is the phenotype, the outward expression of the genes, and this is the genotype, which is the actual alleles that they possess. Once again, just reviewing the terminology. In the review sheet, at the very beginning of genetics, I'm going to basically write something like, okay, when we got to genetics, we threw out a bunch of new terminology, and I'm just going to list a bunch of terms that you need to make sure that you're clear of the definition of. Things like genotype, phenotype, homozygous, heterozygous, dominant, recessive, etc. So then what Mendel always did, Mendel would go and allow these individuals to self-fertilize. So I'm going to erase all of this, and I'm going to write the next form of the cross up here. So we're going to have a heterozygote crossed with another heterozygote. And I taught you last time, how do we get the gametes from this individual?
Is that when we talked about the distribution process thing?
Yep. So we do FOIL, first, outer, inner, and last. So we take the first items of each trait, the outer ones, the inner ones, and the last ones. And that's the gamete types for one of the parents. Well, the gamete types are going to be the same for the other parent. So we can put those across the top. And what we end up with is a 16 square Punnett square. It looks something like this. Now, what's the next step that we would do if we were working this out? Well, we are working this out. What's the next step? You want to combine the gametes, right? How are we going to combine the gametes?
I'm going to go across and pair the top to the side.
Okay, so we can bring these all the way across. So let's bring these all the way across. Notice that I'm giving, since the convention is to put the dominant allele first, I'm leaving space after these for whatever the other allele is. Okay, so I've brought all of those across. Now what do I need to do?
Go from the top and down.
So now bring these all down.
What are you bringing down?
These gametes, these
Okay.
alleles here, are going down.
Okay.
Little r, big Y, little r, big Y, little r, big Y, little r, big Y, little r, little y, little r, little y, little r, little y, little r, little y. So I brought these across, and then I brought these down. So now we've gone from haploid gametes to diploid offspring. So now we have 16 different cells, and these cells have genotypes in them. We can start at this first genotype up here. It's homozygous dominant for both traits, and we have one individual of each of those. As we work our way across, we see that we have this next genotype. How many times does this genotype occur in this table?
Twice.
Twice, yeah.
It occurs twice. And where are they located?
Far from each other?
They're located right here next to one another, right?
Very close. Huh?
So this is homozygous dominant for the first trait and heterozygous for the second trait, and there are two of those. But they occur on this diagonal. So maybe there's a structure to this table that's informative. What's on the next diagonal? Well, on the next diagonal, we have a homozygous dominant for the first trait and homozygous recessive for the second trait, and there's only one of those in the entire table. But then if we look at the remainder of that diagonal, what do we see? We see a heterozygous for the first trait and homozygous dominant for the second trait, and there are two of those. So we've now taken care of this diagonal. What's on the next diagonal?
Four heterozygous ones.
Speak up, Blair. Is that Blair? I can hardly hear you.
Four heterozygous ones.
Okay, you have four individuals that are heterozygous for the first trait and heterozygous for the second trait. And there's four of those. On the next diagonal, what do we have? We have two individuals that are heterozygous for the first trait and homozygous recessive for the second trait, which is kind of just the opposite. It's exactly the opposite of the thing just across the diagonal from it. Homozygous dominant versus homozygous recessive, homozygous recessive versus homozygous dominant. Sorry, I messed that up. It's just kind of, wait, where am I at? Oh, heterozygous for the first versus homozygous for the first, and homozygous for the second versus heterozygous for the second. And there are two of those, and then there's this guy here in the middle that is all by himself, homozygous recessive for the first, homozygous dominant for the second. Then we have this diagonal here, where we have homozygous recessive for the first, heterozygous for the second. There are two of those. And then the last individual down here is homozygous recessive for both traits. So in the same way that we had a genotypic ratio for a monohybrid cross that was a 1:2:1 ratio of homozygous dominant to heterozygous to homozygous recessive, we have a 1:2:1 to 2:4:2 to 1:2:1 ratio of all these different genotypes. So you can have a genotypic ratio of a big cross like this in the same way that you have a genotypic ratio of a small cross. So this is all illustrated for you in this slide.
Hey, I have a question, Dr. Quincy. So can we have a homozygous recessive come before a homozygous dominant? Like as we're filling it out, so in sense, can we have a little y before we have a big Y?
No, the convention is generally you put dominant allele before you put recessive allele. That's just convention. If you did it the other way, I'm smart enough I can figure out what you're meaning, but the convention is to write it this way. And so you notice that while I was filling this in, when I filled in this row right here, when I filled it in originally, as I was writing it, I wrote it with a little r right next to the big Y, little r next to big Y, little r next to big Y, little r next to big Y, because I know that the little r can always come second in whatever genotype you're in, and I know that the big Y is always going to come first in the next trait. So that regardless of what else was there, I could put the ones that I'm bringing down in the right place. I can bring that big R there, and I can put that little Y there. This little r can go in the front just as easily as going in the back. The big Y can go after that big Y because there's two of them, but then r little y as I bring those
Makes
down.
sense.
So because I've done this about 5,000 times, I have my own pattern for how I do it, and I write the gametes in a way that I know that I can fill in the other set of gametes in the correct way. So, but that's the convention. Dominant always comes first in a heterozygote when you write it. But if you do something varying from that, I'm smart enough I can figure it out. So each of these genotypes, though, refers to a phenotype. In this case, this seed is round and yellow. In this case, this seed is round and yellow. In this case, this seed is round but green. This seed is round. This plant is producing round yellow seeds. This is producing round yellow. This is producing round green. This is producing wrinkled green, wrinkled yellow, rather. And this is producing wrinkled yellow. And this is producing wrinkled green. Did I do that right? So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 9 individuals that are round yellow in terms of their phenotype. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we've taken care of those. We have 1, 2, 3, 3 that are round green, round seeds with green color. So we've taken care of those 2. We have 1, 2, 3 that are wrinkled but yellow, and we have 1 that is wrinkled and green. This then is the phenotypic ratio: 9 round yellow to 3 round green to 3 wrinkled yellow to 1 wrinkled green. And that comes out of this Punnett square. And this is the ratio that you're probably, if you're familiar with any of these things, this is the ratio that you are more familiar with. In this case, it's the most common phenotype is the combination of dominant trait for both genes, for both traits. 3 of them are recessive for one trait and dominant for the other. The other 3 are dominant for one trait and recessive for the other. And then the least common is the one that is recessive for both traits. So in 9-3-3-1, one way to think of the 9-3-3-1 ratio is if you have 2 traits, 9/16 of the population will be dominant for both traits. 3/16 will be dominant for one trait, recessive for the other. 3/16 will be recessive for one trait and dominant for the other. So if this is trait 1 and trait 2, you have to flip these, and then 1/16 is recessive for both of the traits. This is another way of thinking of it, regardless of what the trait identity is. Okay. Are you clear on how Punnett square works and how you can work through these things?
Yeah, well, we have to find the ratios.
Yes, you will. So I have prepared for you. Let me go and find it. That's another thing on my list of things to do today. is, I have a practice homework set for you. And it would help if I could spell. So let me share a different thing. So just to get you in the habit of doing this, can you see a PDF now? Is there a PDF on your screen?
Yeah, yeah.
Yeah. So there's a practice homework that I've used in the past that basically gives you some practice doing some genetics problems so that you're equipped to handle them when you get to the test. So I'll send that. I'll put that on Moodle later on today. It's not something for you to turn in for a grade. It's just something to once again help you study and make sure that you understand the concepts. So I'll put that up so you can get a chance to practice some of that. Once again, practice it before we get to class on Friday, and then you can ask questions about it at that point. So this is a simple dihybrid cross. It always results in 4 different gamete types and 16 different types of offspring genotypically, but then there are commonalities in this. And if you make your gametes using the FOIL method, they always get ordered this way, and so there's always this symmetry to the table. It doesn't have to be symmetrical, but once again, I find it easier to remember things when I come up with systematic ways of dealing with the information. And so this is once again just a habit that I've gotten into over the years. In the gametes, dominant recessive, recessive dominant, sorry, dominant dominant, recessive dominant, dominant recessive, recessive recessive. Do that the same way on both sides, and you get the symmetry where you have these diagonals that are kind of mirror images of the table. Now, on an exam, I might actually give you 2 parents and I'll tell you what the genotypes are, or I might give you a phenotypic ratio and say, okay, this doesn't conform to the phenotypic ratio that you see down here. So what's different about the genotypes of the parents that is leading to a ratio that you don't expect? Normally, you would expect a 9:3:3:1 ratio. Well, what happens when you don't get that? We worked a simpler example of that on Monday, where we basically had one of the parents being heterozygous rather than homozygous. And so whenever you get an unexpected ratio like that, you can ask yourself, okay, well, there must be something going on weird with the genotypes of the parents. And there's a couple of problems on that in the homework practice. So unfortunately, well, okay, so here's some of Mendel's actual results. He did an F2, the form of the seed and endosperm color, and these were his results, and these are his ratios, a 9.84 to 3.16 to 3.36 to 1 ratio of these different phenotypes. So they're not very far off of the expected ratio of a 9:3:3:1 ratio. So the unfortunate thing about things like the color of the seed and whether it's wrinkled or not aren't necessarily of interest to people, especially people who raise plants and animals. Because if you're raising cattle, you don't really care what color those cattle are. You're concerned with how many pounds are they carrying around with them, because the heavier they are, the more you get for them at market. Well, as it turns out, those kinds of traits like weight and height and length and even the number of kernels in an ear of corn, those kinds of traits are oftentimes controlled by multiple genes all acting on a single trait. And so there is a genetic term called pleiotropy. Pleiotropy is when multiple genes affect a single trait. Pleiotropy. No, wait, sorry. I'm losing my mind. Erase this from your memory banks. I just made a stupid mistake, I think. Let me go back and check myself. Yeah, I was just the opposite. Sorry. Blah. No, forget that. Pleiotropy. I got them reversed in my head because I'm thinking on my feet. What we're talking about now is a situation where multiple genes affect a single trait. This is what we have when we have additive genetic traits. I don't know why I said pleiotropy. So what happens in these situations is, in your case, a height is a good example of a trait like this. So if we were in class right now, I would line you guys all up against a wall, and I would ask you to sort yourselves from the tallest person to the shortest person. And what you would see is that there aren't big jumps in the height of people. It's not like with peas where you have, you know, tall plants and short plants, and there's nothing in between. You would see that some of you are tall, some of you are a little shorter, some of you are a little shorter, a little shorter, a little shorter, a little shorter, all the way down to the shortest person in the class. And these are traits that tend to be governed by lots of different genes. So there are lots of different genes that contribute to you being a particular height. And in these genes, what happens is each dominant allele contributes a little bit to that, that particular trait. So in humans, there are a number of genes that are like this. Height is one of them. Eye color is one of them. Eye color is controlled by 3 genes. Each gene contributes something different to eye color. Skin color is all determined by how much melanin is in your skin cells. Those are also controlled by multiple genes. Each one contributes a little bit to melanin production. Pasty white people like me have very few dominant alleles that are contributing to that. People with darker skin have more dominant alleles than I do because I'm a pasty white boy. Can't go out in the sun without getting sunburned. So we can think about these traits that are governed by multiple genes with standard Mendelian genetics, the kind of genetics that we're looking at here, but we have to think about more genes than just 1 or 2 genes. And so on this next slide, what I have is a situation where we have 3 genes. So imagine that we have a trait. Let's just say that it's height, and it's governed by 3 genes, gene A, gene B, and gene C. If you have a dominant allele for gene A, it contributes 1 unit of height to you. If you have a dominant allele for gene B, it gives you 1 unit of height. And if you have a dominant allele C, it gives you 1 unit of height. Let's say that if you are homozygous recessive for all of these different genes, you have a height of 1, because everybody has to have some height, but that's the shortest person in the population. If you have a homozygous dominant individual who's homozygous dominant for every gene, you have 1, 2, 3, 4, 5, 6, plus the 1 unit of height that everybody gets. So you have individuals who are 7 units tall. this individual is 1 unit tall. So if you have an individual who has one allele that is dominant, but all the rest are recessive, this individual would be 2 units tall. If you have an individual with 2 alleles, this would be an individual that's 3 units tall, et cetera, et cetera. But it doesn't matter which gene has the dominant allele. This individual is 2 units tall, but so is this individual, and so is this individual. Okay, are you following the rationale? Okay. And these are the only 3 ways of getting an individual 2 units tall, right? So we can do this by building a Punnett square the same way we've done it in the past. So let's take a homozygous dominant individual for all traits, cross it with a homozygous recessive for all traits. It's easy to figure out what the gametes are going to be for each of these individuals. This individual can only produce dominant alleles, A, B, and C. This individual can only produce recessive alleles, little a, little b, and little c, and their offspring produce a individual who is heterozygous for all 3 traits. How tall is this individual?
4 units?
4 units tall. So this individual that is 1 unit tall and this individual who is 7 units tall, when they produce offspring, all of their offspring are 4 units tall. Now, if we take those offspring and allow them to interbreed, which we normally wouldn't do if we're talking about humans, but this is just a class example, we would take these 2 genotypes and cross them. How do we make gametes now that we have this 3rd gene here?
The same?
Okay, so we can start off the same way. We can partition this and say, well, we'll take the first 2 genes and we will basically just do FOIL on them the way we normally would. But then we have to get the 2nd gene in here so we can put big C's on top of that, then just FOIL it again.
That is doing so much.
And then put little c's at the end of that. So now you have all possible combinations of these alleles in the gametes. So now you have 8 possible gamete types. You'll have 8 possible gamete types from that individual. So you have a Punnett square that looks like this. Does that make sense? So now you have 64 possible offspring types. So what time is it now? 9:46. Okay, I'm gonna wait here while you guys work through figuring out all of the gamete types, or all the offspring types. So get to work. I just saw the look on Mary's face. She's like, he's not really making us do this, is he? No, I'm not gonna make you do it. It's ridiculous. It takes so long to work out. So what I've done is I've done it for you. But look at this Punnett square for a minute. So all I've done is I've done the same thing. I've taken these gametes and I've brought them across, and I've taken these gametes and brought them down. What's at the very upper left-hand corner of this Punnett square?
Homozygous dominant.
Homozygous for every trait. What's at the bottom right-hand corner? Speak up. What's at the bottom right?
The homozygous recessive.
Homozygous recessive for all traits. Look at this first diagonal. What do you notice about the first diagonal?
They're the same.
They're the same. What about the second diagonal?
They're also the same. No,
Well,
no,
this one,
not the same.
go ahead, Marcus.
No, I was saying like they're not the same.
This one is the same as this one, and the one in the middle is different, just like it was in a dihybrid cross. What's in the long diagonal going all the way across this table?
The two corner ones are the same.
Two corner ones are the same, but what about all the ones in between?
They're all the same.
They're also the
They're all the same. So those are all of the heterozygous. Those are all heterozygous for both traits. So because we're talking about height, and each dominant allele just adds a little bit to your height, does it matter what the individual genotypes are, or does it matter how many dominant alleles you have?
The dominant
All that matters is the number of dominant alleles. So if we look at a table where we've basically taken these genotypes and said, how many dominant alleles do we have? You'll see that it starts high at this side, and as you go diagonally across the table, you get fewer and fewer and fewer dominant alleles. You'll also notice that the top row goes 6 dominant alleles to 5, 5, 4, 5, 4, 4, 3. What does the first column do? 6, 5, 5, 4, 5, 4, 4, 3. That symmetry is still there just as it was in the other one. So this individual has 6 dominant alleles. What would its height be?
7.
7 units tall, 6 units tall for these individuals that have 5 dominant alleles. So whether we count the number of dominant alleles or whether we translate this into height, what do we notice here? How many individuals have 7 units of height? Because they have 6 dominant alleles.
1.
Okay, so there's only one of those. One. I need to get some new markers, I think. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 15, 16, 17, 18, 19, 20. So we only have one individual. How many individuals do we have with only one unit, which is 0 dominant alleles?
One.
Okay. How many individuals do we have with 2 dominant alleles? Sorry, 1 dominant allele with a height of 2 units.
4.
I see
Okay.
1, 2, 3, 4, 5, 6. I see 6 individuals with 1 dominant allele and 2 units of height. So 6 is right here. How many do we see with 2 dominant alleles that would be 3 units of height? 2 dominant alleles. I see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. I see 15 of those. Three dominant alleles and 4 units of height is the midpoint of this distribution. How many individuals do we have with 3 dominant alleles? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Four dominant alleles, height of 5. How many would we expect to see? You can count them up, but you don't need to count them up. Why do you not need to count them up?
Because it's inverse of the, or it's a reverse of the previous graph.
Yeah, but because the Punnett square is symmetrical, this distribution will always also be symmetrical. So we should find 15 of these. We should find 6 individuals that have 5 dominant alleles with a height of 6. We should expect to see 6 of those, and then I'll erase this and bring this in to make it seem a little more normal. 7 units with 6 alleles should be one of those. And we can check ourselves. Let's go and count the number of individuals with 4 alleles. 1, 2 in the first row, 3 in the first row, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So it works out. What does this thing start to look like? Starts to look like a bell-shaped curve, doesn't it? It looks like this. So when we look at human height, we see that there are very few really tall people, very few really short people, and there are a ton of people right in the middle. And it's because all of those average sized people come from this middle section of the Punnett square where there are lots of 2s, 3s, and 4s. Very few 0s and 1s, very few 5s and 6s, which leads to this normal distribution of traits. And this is commonplace. IQ scores follow a normal distribution. Height follows a normal distribution. Weight follows a normal distribution. Oh my gosh, so many things follow a normal distribution when we think about them in terms of skin color follows a normal distribution, et cetera, et cetera. These are the kinds of traits, though, that in many cases are of interest to farmers because they're interested in how many kernels of corn there are on a cob of corn, because that affects how many bushels of corn they can produce. That follows a normal curve. They're interested in how many gallons of milk their cows produce. That also follows a normal curve. Very few cows produce a lot of milk. Very few cows produce very little milk. A lot of cows produce an average amount of milk. Well, it turns out that these kinds of traits, because they're determined by individual, a number of different genes, each individual allele contributing a little bit to the phenotype, these genetic traits are actually very easy to do breeding with because they have this quantitative nature to them. But remember that this distribution, this normal distribution, came out of simple Mendelian genetics. It's just the Mendelian genetics of multiple traits acting on a single gene. So this is once again corn color, wheat color rather. In a wheat grains have different colors. You can have white wheat, you can have dark red wheat. And once again, these are controlled by 3 different genes, and you end up getting this distribution of wheat color in the seeds. Well, as it turns out, this is cool because rather than doing big Punnett squares like this, we can actually rely on the fact that these traits have a distribution, and rather than analyze them with kind of more traditional genetic methods, we can analyze them just by looking at the phenotype. So one of these traits that is a trait that is quantitative is height in humans. And so the way you figure out whether or not a trait is heritable and how heritable it is by looking at the phenotype in the parents and comparing that to the phenotype in the offspring. So this whole thing here is a parent-offspring regression. So this is a group of 64 undergraduate students that were in my evolution and ecology class a number of years ago. And I asked them to go home and get the height of their parents and to get the height of all of their siblings who had grown up. If they had young children who were siblings, we excluded those because they haven't reached their overall height. Well, as it turns out, if you do a regression between average offspring height and parental height, the slope of this regression, which in this case is 0.91, is a measure of heritability. And let me just define heritability for you in kind of plain, simple terms. Heritability is the degree to which the degree to which parental genes affect the phenotype of the offspring. And the way we measure this is by doing what we call a parent-offspring regression. To what degree is the phenotype of the parents correlated with the phenotype of the offspring? If the genes in the parents have nothing to do with the phenotype of the offspring, the slope of this line will be 0. Really tall individuals produce average sized offspring, really short individuals produce average sized offspring. But what happens in reality is tall parents tend to produce tall offspring, short parents tend to produce short offspring, average sized parents tend to produce average sized offspring. That's because the genes that determine height in parents also determine the height of their offspring. And so this simple regression of the offspring phenotype with the parent phenotype tells us kind of what percentage, if you want to think of it this way, what percentage of offspring height is determined by parental genes, in a sense. So the reason that this is important, knowing how heritable a trait is because if you want to breed cattle that will give you greater milk production, if this was parental milk production and offspring milk production, you wouldn't want to breed these individuals down in this part of the distribution, would you? Because you're going to have low productivity parents producing low productivity offspring. You want to actually prevent this part of the distribution from breeding because you want the most productive parents breeding with one another so that you get the most productive offspring out of that breeding effort. And this is actually the basis of plant and animal breeding. So if we take this same figure, well, let's see. Yeah, let's leave it here for now. So between now and Friday, take a look at this figure and what we've done up to this point and make sure that you understand this. What I'm going to show you on Friday is how we can use heritability to predict what degree of change we will see from one generation to the next generation as we, for example, selectively breed a group of animals for, more weight on the bone on cows that you're raising for meat production, or more milk production from dairy cattle, or more seeds being produced by soybeans or corn or things like this. But we're going to use this fundamental relationship to illustrate that. Questions about this? All right, then look for some things popping up on Moodle later on today, and I will see you guys all on Friday.